In this video, we will discuss about this microglial depletion can restore these cognitive functions. So far, we have understood the basic mechanism for cognitive impairment in the CAR T cells treated patients is because of the formation of these microglials. So they wanted to check if researchers wanted to check if they can deplete this microglial, then whether it will restore the cognitive functions or not. So they checked three different models. First one is this diffusive intrinsic pontine glioblastoma, acute lymphoblastomia, and aggressive osteosarcoma. And this is the and for all these three models they have slightly different timeline. So first one, when they use this DIPG xenograft, then after 8 weeks they started this CAR T cell treatment on target CAR T cell treatment, and from this 21 to 35 days they introduced this microglial inhibitors, that is CSF1R inhibitions, CSF1R receptor inhibitions using CSFI. And for this acute lymphoblastic leukemia, the after all xenograft models, after 3 days they started using CD19 CAR T cell treatment, and from 14 to 28 days they used this CSF1R inhibition, which is mainly responsible for white matter macroglia, and they got these informations from the previous literatures. And for this aggressive osteosarcoma, they use the xenograft, and after that, after 7 days, after that day 7 days of this xenograft inductions into the mice, they started CD19 CAR T cell treatment, and from 21 to 35 days they used the CSF1R inhibitions. So when it comes, and for these DIPG models, they are able to see that the macroglia formations using this marker IBA1 plus marker shows that this macroglia formations goes down in the case where the CSF1 inhibitors is used for both MOC T cells control and also for this on-target GD2 CAR-T1 cell therapeutic approach. And when they did it for this acute lymphoblastic leukemia model, in this case also they can see that with this CSF1R inhibitor, they see a decrease in the microglia formations, and similar results they have observed for osteo or osteosarcoma. Compared to the vehicle, when they use CSF1R inhibitions, they see a decrease in the microglia formations. When they are able to see its impact on this oligodendrocytes precursor cells, they can see the oligodendrocyte precursor cells restores whenever these CSF1R inhibitors, whenever this CSF1R inhibitions is been done using CSF1R inhibitor for both MOC T cell control and also for the GD2 CAR T cell therapeutic approach. And similar kind of results they have seen for acute lymphoblastic leukemia and also this aggressive osteosarcoma for these oligodendrocytes precursor cells. And for these oligodendrocytes glial, this oligodendroglial, and these oligodendroglials are also restored in all the three conditions whenever they use in all the three models whenever they use CSF1R inhibitors, and both of these things, that is oligodendrocytes precursor cells and oligodendroglials, goes down with white matter macroglia formations. So when they suppress these macroglia formations using CSF1R inhibitions, they can see restores in these activities. Thereafter, the final check they did using the cognitive test using this NORT, and they're able to see that with NORT in the CSF1R inhibitions, results is in line with the vehicle, that is when they use CSF1R inhibitions, the recognition ratio is almost similar to what they have seen for the vehicle, and same results they have seen for these on target GD2 CAR T cell therapies. In this case, they can even see the upregulations of these recognition ratios from the CSF when they use CSF1R inhibitor compared to the case when they do not use CSF1R inhibitor. And similar results they have seen for this acute lymphoblastic leukemia and also for the aggressive osteosarcoma. Although the CSF1R not controls 100% of this white matter macroglia formations, but it controls majority of these white matter macroglia formations. So this helps in by targeting this white matter macroglia using CSF1R receptor inhibitions, they can restore some of this cognitive functions that is validated using the NORT test. So I hope you understood how they finally validate their previous findings that microglia is causing the cognitive depletion in the CAR T cells treated patients. If you need to understand more about this experimental results and their approach in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.